This Week in Virology, the podcast about viruses, the kind that make you sick. From MicrobeTV, this is TWiV, This Week in Virology, episode 1309, recorded on March 27th, 2026. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Madison, New Jersey, Brianne Barker.
Hi, great to be here. In Madison, it is currently 50 degrees Fahrenheit and a bit cloudy, which is 10 C, much cooler than it has been the last couple of days. But at least it's not, winter.
So, it's 10 C and cloudy here, too. How about that?
It's shocking.
Tomorrow it's going to zero again.
Mm-hmm. But yesterday it was like in the 70s.
I know.
So
it's crazy stuff. Yeah, it's going to get back up into the 70s. It goes up and down. It goes 70s, like 10, 4, 11, 2. I guess that's spring. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. 85 degrees, and I guess it's partly cloudy. And tomorrow, we're still in sort of crazy season here. Tomorrow it's going to be a high of 69. That still probably sounds pretty warm to you guys. But we're, anywhere from 70 to 90 degrees over the next 10 days.
And from Western Massachusetts, Alan Dove.
Good to be here. It's, I guess I've got the cold temperature today. It's 42 Fahrenheit, 6 C, and cloudy and a little breezy. A front blew through. And so we've got that, and down below freezing again tonight.
Congratulations on your judge who told RFK Jr. he can't do what he's been doing.
Yeah.
Massachusetts is a pretty cool state, isn't it?
It is. It's got a lot going for it. I mean, It's got some downsides too, but
it seems to be politically okay, right?
I mean, we've got the political pathologies that a healthy democracy should see.
Okay.
There's a certain amount of that you expect, and you say, "Oh yeah, it's politicians doing that thing again." But it's at the normal range.
So the upshot is that he said the ACIP has got to be disbanded because you're supposed to use experts. It's a federal law.
Yep. And
all the vaccine changes, you made them yourself. The committee's supposed to do it, so they're all null too.
Arbitrary and capricious. Yep.
Very nice. And what's the other thing that happened? Oh, yeah. So they're not going to contest it. They're going to just name a new committee. Let's see what other bozos they can pull out of the woodwork.
Right.
Right? I mean, there must be a limit to the number of bozos, no?
Yeah, like at some point, shouldn't they run out and have to
run out of bozos?
No, you will never run out of idiots. I'm sorry.
Right.
That's just not going to work.
Oh, gosh. Okay. Well, by the way, if you enjoy these science programs, and they're mostly about science, we'd love your support. Microbe.tv/contribute. Don't forget the Rosenfeld Lab has moved to Philadelphia. They're looking for a postdoc and a technician to work on enteroviruses. They are interested in cross-reactive immune responses. They are interested in animal models and making new vaccine platforms. You should email Amy if you're interested at rosenfelda@mlhs.org. ASV is coming up, the annual meeting in July 27th at the University of Minnesota in Minneapolis. You can go to asv.org and find the program, information about child and dependent care, information about digital access, and registration. Early bird is open until June 1st.
And if you're flying there, you should get on the TSA line now.
Didn't he say he was going to pay them?
Supposedly they're going to deal with that. It's kind of.
I hope so, because I'm flying on Thursday to Kansas.
There was something passed the Senate, a bill passed the Senate this morning, saying to pay
the TSA,
pay HHS except for
ICE.
Except for ICE.
And what about the voter limitation bill? Did that go through?
That's a separate deal. That's not,
good,
as far as I know, that's not happening. Okay.
I was hoping they would not cave on that because that's really improper.
Yeah.
Right?
Yeah.
Oh, my gosh. A few people have died who were scientists. Well, Mike Bishop has passed away. Of course, he and Harold Varmus co-discovered SARC, the first oncogene to be discovered, and they both got a Nobel Prize for that. We had, of course, Harold Varmus on TWiV, but not Mike Bishop. But I did interview him years ago, and we'll put a link to that interview in the show notes. And also, David Botstein has died. He was not a virologist, but a very good scientist, a very smart guy. And you always knew when he was in the room. He was very loud and not afraid to talk. And he's famous for using restriction fragment length polymorphisms to map genes, and then later to figure out how to properly use DNA micro arrays. I was reading his obituary, and unfortunately his brother, who's president of Bard College and head of the orchestra, is in trouble over Epstein stuff.
Oh no.
He got money from him, and he went to his island, and it's not good. And so people are calling for him to resign, which I think he should. He should not be president of a university, having gone to the island and done stuff with Epstein and taken money from him. So just like with Richard Axel, he stepped down as head of something. He gave back his Hughes money, and he didn't even go to the island. He just said it was a bad thing to do.
I have a couple of anecdotes about these dudes.
Not Epstein.
No. I knew neither Mike Bishop nor David Botstein Wells. Just, ran into them a couple of times. They certainly wouldn't know who the hell I was. Mike Bishop. First of all, I remember I immediately go to Gordon conferences where sort of the standouts in terms of talks were Bishop and Varmus and Baltimore. And Mike Bishop always gave absolutely the coolest talks that were laced with all sorts of non-science stuff, literature and art and everything else, pithy quotes and stuff. He was just amazing. When I was a lowly postdoc, after my first year of postdoc, I had, for other reasons, a visit to the U.S., and I was encouraged to contact people and do a sort of a seminar tour, even though I didn't have anywhere near the kind of stuff that would be suitable to give a seminar. And I wound up going to UCSF, and Mike Bishop hosted me. I don't know how that happened, but I visited him in his office, and he took me around to my seminar and that kind of stuff. And he was just absolutely so welcoming and so wonderful. He didn't have to do that. I was still am nobody. And I really appreciate that. The other thing was he interviewed for a chair's job at Stony Brook while I was there when the chair was vacant. Okay. Nobody thought he would actually seriously take the job. But the great thing was he wore canary yellow socks with in these high water pants. So the canary yellow socks really stood out. It was awesome.
And you mentioned him at the Gordon conference. I remember I saw him many times. He used to read his talks. He wrote them out, every word.
Oh, okay. I guess I didn't realize that. Okay.
Yes.
So, and the David Botstein anecdote, I got this third hand, but I believe it. Second hand. He is supposedly the author of a quote that is, "The ability to do genetics is an inborn error of metabolism." Which is absolutely true. Genetics is a special way of thinking. And David knew that because he had that mutation.
It's cool. I have just one story here. I originally found it in the Times, but I have an AP version because it's open. Trump administration opens investigations into race in admissions at 3 medical schools. So we have Stanford University, Ohio State, and the University of California, San Diego. All right. So this expanding his nonsense.
Yeah.
This comes under the general heading of bullying.
Yeah. Yes.
Mm-hmm.
Well, it's also, it's bullying, yes, but the Supreme Court said you can't do affirmative action anymore, so they want to see if these universities are still doing it.
But it's just like the bullying of other institutions with sort of thinking about their undergraduate admissions.
Yeah.
It's coming
they
more
want
broadly.
admission. They want admission records to start with. So there you go. Okay, now we have some science, very interesting science. I found these a couple of weeks ago, and we're finally getting around to them. Now, the first paper is in NPJ Vaccines, Post-Attachment Neutralization of HPV, Human Papillomavirus 16, by antibodies derived from Gardasil-vaccinated women.
The
And Alan?
first author is Patricia Day. Senior author is John Schiller. And the team is from the National Cancer Institute at the NIH in Bethesda, Emory University School of Medicine in Atlanta, and the Fred Hutchinson Cancer Center in Seattle.
Now, the reason I did this paper, I found it because a few weeks ago on my live stream, people say, why is the HPV vaccine sterilizing? And I say, I hear that all the time, but I've never actually looked into it.
We have to be careful here.
Yeah, of course.
The term sterilizing immunity for a vaccine that's given to prevent a sexually transmitted disease is a very, very unfortunate choice of term.
I agree. It blocks infection.
Why is it so effective is the question.
But unfortunately, the term sterilizing is in the lexicon.
It is, yeah.
Well, and there is an important distinction between that mechanism and other mechanisms that we have been discussing for eons, and I was really happy to see this paper because it addresses it head on and really validates a lot of the discussion.
I always, to me, the literature says vaccines prevent disease, but they permit infection because a memory response is a few days delayed, and therefore the virus can get in and replicate. Of course, if you have very high levels of antibodies or T cells, or say early after vaccination, maybe that could prevent infection, but certainly longer it wouldn't. So I said, okay, I'll look in the literature and then I'll come back and give a little mini lecture, which I did. And I found this paper, and holy cow, it is the only vaccine that prevents
infection.
Because then this week I did it for measles vaccine, and it's very clear that it does not prevent infection.
Yeah, I think it's sort of the cool thing is that it's preventing against infection long term.
Yeah.
This was a big topic of discussion during the pandemic, and it confused me as well because, in fact, in my mind, I just kind of assumed that a lot of vaccines were sterilizing. Okay, and then what happened was that people got vaccinated and they got sick. And so everybody throws up their arms and says, the vaccine doesn't work, okay? But they didn't realize that, so you got sick, but you didn't die.
But you die?
Didn't go to the hospital, okay? So then we started discussing, okay, well, what is this? Well, most vaccines don't prevent infection, as you just said. And then we started wondering, are there any sterilizing dudes? HPV was reputed to be that, and here it is. This is great.
Yeah.
I have another.
So, yes.
I got to say, too, this is another anecdote. John Schiller. I don't think I've ever met him. I may have, but I've interacted with him because occasionally I needed material of one sort or another for talks on HPV. And he's just right there, okay? Responds to you immediately and gives you everything you want and more. So thank you, John. This is great.
Yeah. So I, just like Rich, kind of originally had the idea, vaccines, sterilizing immunity. Sure. And I also came to be thinking about this because people were not necessarily having disease after having been vaccinated, but were testing positive for infection. And so I sort of came to a lot of this as, so I guess the reason for this mistake, the reason why we thought they were sterilizing is because we used to measure infection based on symptoms. And now that we got all these fancy, smanchy ways of doing PCRs to actually look for low amounts of virus, we see that actually infection is happening. And so I kind of, when we started talking about this, in particular with COVID, with breakthrough infections, sort of got this idea of sterilizing immunity no longer a thing.
Right.
And so being able to see there are situations where sterilizing immunity long-term is a thing and how it works is so cool.
Yeah.
The other thing that we discussed occasionally is HIV, because this is where this topic comes up. Because in the case of HIV, which just establishes lifelong latency, if a vaccine is going to be effective, really effective, it needs to be sterilizing. So you can't even establish latency. Whether or not that's going to happen is another story.
All right. So we've talked a bit about HPV, human papillomaviruses, recently. These are viruses, human, obviously. They cause warts, and a subset of them are sexually transmitted and can cause cervical and anogenital and oral mucosal cancers. And so vaccines have been developed which are great at preventing those cancers. In fact, if you give them to kids before they become sexually active, they can be 100% effective at preventing cancers. And thousands of women mainly used to die in the U.S. of HPV cancer. So this is a great thing. And the vaccines, one of them is called Gardasil, another one is Gardasil 9 with more genotypes. These are virus-like particles made in yeast or insect cells. So there's no genome. It's just protein. It's the major capsid protein. It's called L1. And they have, as they say in the paper, they have exceptional potency at preventing cancers. But they say unexpectedly, they not only protect from cancer, but they routinely produce, and I'm sorry, Alan, they produce sterilizing immunity. They actually use the term themselves. Yes. In that, here's the thing, in that most vaccinees never test positive for the targeted types in DNA amplification assays. So they have a citation there. So I went to the paper to see. And the paper that they cite is a 2011 paper, Prevention of Persistent Human Papillomavirus Infection by an HPV 16-18 Vaccine, a community-based randomized clinical trial in Guanacaste, Costa Rica. Now, it's not as good as they say, because vaccine efficacy, and they're looking at infection by PCR here, because it's very hard to grow the virus. 90%, right, against 16 and 18. Okay, so not 100%, but 90%. But at year 4, it's 100%. It goes up.
It gets better.
It actually gets better, which is incredible. And so this is the other feature of this immunity. It's incredibly long-lived. And I've read elsewhere that the plasma cells that produce these antibodies just live for ages. And
does anybody know why? I ran across this too. Immunologists, it's a jaw dropper for immunologists, okay?
Yeah.
That the antibody levels stay so high for so long. Anybody know why?
No.
I don't know why.
Okay.
No. So they say, okay, so there's consistently high levels of antibodies for more than a decade, right, as long as you've been studying this. But they say there must be some virology factors. So that's what they're going to look at in this paper. And so the way this virus infects is very interesting. So in the cervical region, you have a mucosal epithelium consisting of multiple layers of cells, with the ones on the top the most differentiated, and the ones at the bottom, the basal keratinocytes, are the least. And the virus needs to enter the basal keratinocytes, and it can't do it unless there's a break in the mucosa. So they say here they have a mouse model where viruses cannot get in to intact columnar epithelium. And so the reason this is that those basal keratinocytes, the virus attaches to the basement membrane, actually, which is under the basal keratinocytes. They attach to heparan sulfate proteoglycans, and that binding induces a conformational change in the capsid that exposes the minor capsid protein L2. It then is cleaved by furin, a protease. It undergoes additional conformational changes to expose a site on L1 that then binds a kerasinetyte receptor. And then the virus can start getting in. But all of that takes hours. And so that's a window where antibodies can work as they
and the reason the virus probably evolved this convoluted mechanism and this difficult to reach target cell is probably because it relies on the differentiation of the keratinocyte to run its life cycle. And also, if you just infect the superficial epithelial cells, they're just about to be gone anyway. So this is a much better target for it, but that means it's got to get all the way down to the literal basement and then go through this multi-step process to get into just the right cell.
So I always knew that the virus infected basal cells through a wound, but this other stuff of having to bind the basement membrane and go through these conformational changes to make it capable of infecting those cells, that was news to me, and it's fascinating.
And I didn't realize it was so slow at it.
Yeah.
Yeah, I didn't know that either, because usually uptake is pretty quick.
Yeah.
Yeah, I mean,
for
most viruses, it's like you bind, you go in.
This is actually a good point here. You know, you learn things with model viruses, and you assume they apply to everything.
Yes.
Right? Viruses getting quickly
They're all
by
fast.
endocytosis.
They're all lytic. They all, yeah.
But this is, you have to keep your mind open, right? Anyway, so it was known before that if you give say mice, as a mouse model, high levels of sera, it will actually prevent binding of capsids to the basement membrane. But if you use lower levels of sera and lower antibodies, the capsids can bind. They can undergo the initial conformational changes needed to get furin cleavage, but there's no entry. And so, in fact, in this case, the capsids are ending up in phagocytic cells, so they're probably grabbed by those cells. So the antibodies are bound to the viruses, and the phagocytic cells take them by Fc receptors. But they want to know in this study what happens to viruses that have completed these conformational changes. They're bound to the keratinocytes. Now, classically, a neutralization assay, you take virus and antibodies or serum, and you mix them, you incubate them, and then you put them on cells. And this has been done before with 293 T cells, which are biologically irrelevant to human papillomavirus infection, but you can study neutralization. And so here what they're doing is using a human keratinocyte cell line. It's a surrogate for basal keratinocytes. It's called HACAT cells, HACAT.
Or
And they have
Yeah, I always call it Hackett.
Hackett. They have sera from Gardasil vaccinated women, and they also have a panel of monoclonal antibodies against these virus-like particles that have been derived from memory B cells from vaccinated people as well. Okay, so first they say, do these neutralize in the normal assay? Mix it with virus, add it to cells, and 9 of the 13 monoclonals, they prevent infection. So they say, let's do it in a cell line with greater relevance for HPV biology, which is funny because when I was starting to study innate responses to poliovirus, I was using HeLa cells, and people said, why are you using HeLa cells? They are not relevant to poliovirus biology. And I said, you're right. Okay, so now these cells, if you do a traditional neutralization assay, it works. But then they want to do a different kind of assay where they allow viruses to attach to the cells. Excuse me. And then they add the antibodies. And then they measure infectivity at different times after addition. All of these antibodies can neutralize by infectivity, we're looking at the virus getting into cell and replicating. They all neutralize still at 3 hours post-attachment. And then there are a couple of non-neutralizers that don't do that as well. So then they went longer. They went up to 18 hours. And in fact, they still get some inhibition of infectivity at 18 hours post-attachment. This is really amazing, right?
Yeah.
Usually you don't see that.
Why is the virus still there?
And there seems to be some differences in these individual antibodies, and they say maybe there are different mechanisms of post-attachment neutralization, and that's what they study in this paper. Okay, so then they as I said, these particles undergo conformational rearrangements, and so they wanted to make sure that these antibodies will recognize the virus particles at different parts of this process, so you're not losing it because of the conformational changes. So they can all recognize the cell-associated papillomavirus. virus-like particles, with the exception of one antibody. And then at 5 hours, you can, for all the antibodies, you continue the 5 hours, you see a perinuclear signal for all the antibodies. So somehow that's a post-attachment neutralization. And at 24 hours after infection, all of the antibodies, with one exception, show very specific staining of viruses in a perinuclear pattern.
Yeah, and so this is sort of completely unlike what you see in the immunology textbook, where you see antibodies doing neutralization always in an extracellular fashion, and the antibody is always outside of the infected cell. And so here the antibodies have bound to the virus and are inside the infected cell still doing something. There are certainly some other examples. I think that at a previous time I mentioned something like this in terms of the classic way it worked. Somebody wrote me an email to try to tell me about some of the other ways that antibodies can get inside of cells. So yes, there's this protein called TRIM21, which they talk about in this paper, where sometimes we do see antibodies inside of cells, but usually, classically, we think about them acting outside of cells in neutralization. So the fact that they're seeing these perinuclear signals and other signals inside of the cell with the virus is really unique.
So the next series of experiments, it's kind of a control. So the virus will also bind to extracellular matrix, and then it gets transferred to the cell, but you don't need to bind to the matrix. So they want to make sure this is not misleading them, and it's not. So I'm not even going to go into that, because don't worry about ECM binding.
Well, let's make the distinction here. You may have already said this, but I emphasize that that's peculiar to this experiment. In vivo, they have to bind to the extracellular matrix to trigger the conformational changes, or at least the basement membrane, which I think of that way. But in this model, that heparin sulfate is on the cultured keratinocytes, where it's not in the basal cells ordinarily. So you don't need the extracellular matrix in this case.
Right. But they're worried that, the antibodies are preventing association with the ECM, and they showed that that's not the case. So then they want to know, so there's pre-attachment and post-attachment neutralization going on here. So they want to know the mechanisms of both, right, for each of the antibodies. So they add the antibody and the virus-like particles together to the cells 3 hours, and then they remove the unbound complexes, and then they fix them, and they look for virus particles with antibodies. This is mostly an immunofluorescence study. So they find 3 different categories for pre-attachment neutralization. So one is that there is cell surface retention of the virus particle throughout the 24-hour incubation period. In other words, you add the virus and the antibody to cells, and it just stays at the cell surface, never gets inside. Of course, that would block infectivity, right? Second phenotype, the virus binds to cells, and then the signal goes away. And then the third category, there's no cell-associated activity at any point, okay? So presumably that's preventing binding entirely. And the first one is allowing binding, and somehow it's interfering later on. And what was the second one? The second one.
No, the second one is interfering later on. Later on, the first one is right. Okay.
And the second one is slowly getting inside, but it's being neutralized.
But it's being neutralized.
And then there's post-attachment neutralization. And so here you allow the virus to attach to cells for 3 hours. You remove the unbound virus, and then you add antibodies for 4 or 21 hours. Right, and then everything is being done by immunofluorescence here. And for some of the antibodies, you see similar levels of virus particles on the cell surface. And then for other antibodies, you don't see them on the cell surface, and you don't see them inside the cells either. And so the loss could be shedding of virus or degradation of in an intracellular compartment. So in fact, these antibodies probably get into cells with the virus and cause degradation. And that's a mechanism we talked about years ago on TWiV. Some adenovirus antibodies neutralized by getting inside the cell with the virus particle, and then they're degraded in what's called a TRIM21 mediated degradation process. I don't know if anybody remembers that paper. It was a long, Rich, you remember it? Long time ago. And so they want to know if this is happening here, but there's no evidence that TRIM21 is involved with this.
Yes. And if you had asked me before I read this paper about neutralization happening intracellularly or any kind of anything basically other than extracellular neutralization, I would say the answer is TRIM21 and that's it. And so now they've shown TRIM21's not what's going on here. So to me, everything after this is completely novel.
And then the other post-attachment mechanism is the virus is binding to the virus, that's the antibody at the cell surface, and then it falls off. So the antibody induces shedding of the particles from the cell surface. And some of the antibodies do that. So they have a couple of mechanisms of post-attachment neutralization. One is that the antibody pushes the virus off the surface, right, at some point. And another is that it brings it inside and it gets degraded. And what's the third one?
It's the retention on the cell surface.
Retention.
Sits there with the antibody.
Just sits on the cell surface,
Yeah.
right? And the different antibodies do them in different combinations, right? Some of them do 1 or 2 or 3 or really.
And
really
one of the
nice.
things that I had to remind myself of, and they get to this later in the paper because we're going to do patient serum, is that they're doing all these studies with monoclonal antibodies. Okay,
Yes.
so you're looking at one
Right.
mechanism at a time. But in somebody
Right.
who's been vaccinated, they're going to be polyclonal. So all of the above potentially
Right.
apply and probably
Right.
do.
Right. So they have now the polyclonal sera from vaccinated women, and they have controls, of course, that have never been vaccinated, and then they have people who, and they're seronegative, and they have people who were infected, and they vaccinate them, and they can get serum before and after vaccination to see what's happening there as well. And they have, I don't know, it looks like about 10, 2, 3, 4, 5, 6, 7, 8, 9 different antibodies. No, those are the monoclonals. Anyway, doesn't matter. Anyway, they all neutralize in 3 hours, within the 3-hour time course. But what are the mechanisms, right? They find that blocking attachment predominates. So they can see that the antibodies prevent binding to the cell surface. And they find that one of them has dissociation. And in fact, what's interesting is that one lady, her pre- and post-vaccination serum shifted in terms of the mechanism of neutralization. So was it before the serum blocked, and then after the vaccination, it shed it from the surface. So you have all 3 mechanisms reflected in the polyclonal sera as well. Right. So basically, what they find with the monoclonals also applies to the polyclonal sera as well. So all of this takes time, as you can see. There's a long window for getting neutralization, and that I don't know how that relates to because they say this is probably a part of the ability of the vaccine to block infection. I guess if you have a long time that you have a big window where you can block infectivity, that's good.
Yeah.
Right? Because they say these are mechanisms, right? We can prevent infection hours after interaction through multiple mechanisms. This is why the vaccines are probably so efficacious, or some new insights into why they're so efficacious.
Yeah, so it may be that this is due to some of the unique biology of the virus and
Right.
how it is infecting, and is not necessarily due to just the antibody responses. Like maybe we can get sterilizing immunity because the virus has this unique way of transferring between cells and these unique sort of steps of infecting in vivo. And so I think it would be a really interesting thing to kind of follow up and try to figure out, because we've mentioned the plasma cells making lots of antibodies that last for a particularly long time. And so how much of this is sort of specific to the antibodies being really good or the antibodies having unique features, and how much of this is specific to the virus? And I was trying to brainstorm earlier how I might sort of investigate those differences of, is it the antibodies are unique or is it the virus is unique? And I didn't get a great answer yet. So
Brianne, how long so the cells that actually produce antibody, they're plasma B cells, right?
Mm-hmm.
How long do they ordinarily last?
Years.
Years.
There are subtypes of plasma cells that I think people are describing more and more in terms of the long-lived and short-lived and plasma cells in different anatomic locations. And
are these, over that period of time, are they regenerated? Is there some stem population that can know?
So that's part of that whole long-lived versus short-lived plasma cell.
Mm-hmm.
The idea is that when a cell has differentiated all the way to being a plasma cell, that's sort of it. There's absolutely no way for it to divide again or become anything else. It just basically produces antibodies and has lost the ability to do anything else. But there do seem to be some memory cells that have the ability to differentiate into plasma cells, and exactly, which of those are particularly important in protection in any viral infection, the short-lived and the long-lived, and this memory cell and that memory cell is sort of the place where immunologists are trying to figure things out right now.
So this is a crazy idea, but you can dump on it if you want. I'm thinking of this long-lived antibody response in these cases. And I'm wondering if there isn't some sort of boosting going on, because there's hundreds of different papillomaviruses. Now, they all have different specificities, so that makes me wonder if you were to look at a person's HPV serum antibody profile over years, whether the specificities would be the same. I don't know.
I mean, you might be able to look at that over time.
There could be boosting. Why not? Yeah.
Yeah.
Why not?
Yeah, that's what I was thinking with this. The longevity of this response was like, is it just like recurring encounters that keep boosting this? Or
it's a good question, right?
Or is there something unique about this antigen that just causes the immune system to become obsessed with it?
Yeah, which would be interesting if you could put a different antigen in the capsid and see if it does the same thing.
Right. If it does the same thing.
Well, or the other thing is there something unique about the vaccine modality? And so, unfortunately, I'm not sure that you could make, say, an inactivated or an alive attenuated, because you have to be able to grow this virus first. But is it something about the VLP and the protein production in terms of how the antigen is produced? Is it the antigen? Is it the virus?
So you could probably make an mRNA vaccine
as your control.
Oh, that would be interesting to see. It may not be as long-lived.
May not be. That makes me think of other VLP vaccines. The first one that comes to mind is hepatitis B, but that's going to be different, because I've always tried to imagine that as a VLP, because it's a membrane virus and you're using the surface protein that's ordinarily in a membrane, but I don't think that's part of the VLP.
It could very well be that hep B in a liver cell is just endocytosed really quickly. And so there's no 4 hours or 5 hours of window. So maybe this is the unique biology of it here.
Yeah, I mean, right. Another possibility is that maybe we generate this type of response to other vaccines that we get, but it just the cells don't have the opportunity to exploit there, because most of those viruses are not dawdling around on the doorstep for hours.
Well, it sure is good news.
Oh, gosh.
Okay. First of all, I'm glad to have...
Now I'm convinced.
I'm glad to have the science resolved, and it also really reinforces how good these vaccines are.
Yeah.
And by the way, they're totally safe. There's no serious side effects whatsoever.
Well, and I think the questions we're pointing out here are all really cool, fundamental science questions that hopefully somebody can address at some point.
So back to Rich, you mentioned, boosting with, you don't need it for long-term memory, right? Faroe Island experiment, where 65 years between measles coming to the island, people still, you have an outbreak, and then 65 years later, you have a second outbreak, and all the people that survived the first outbreak don't get infected. on the second one. So 65 years can last without any boosting.
Right.
And that's the whole point of that island situation, right? Now, it doesn't say that you can't have boosting and that would help, but it's not necessarily in at least the way the biology works. So, but there are other viruses they point out that you get post-attachment neutralization with, and so that's interesting because then you can ask, do they have effective vaccine protection? But we have dengue virus, SARS-CoV-2, Coxsackie-A, herpes, hepatitis C, cytomegalovirus, and chikungunya virus. So, well, SARS-CoV-2, I don't know how durable memory is. We don't know yet, right? It's not been long enough.
Well, I guess the thing that gets into this also that makes it complicated is that this vaccine seems to be pretty antibody driven.
Yeah.
And some of these other types of vaccines we're talking about have both a T cell and an antibody response. And so how you would separate out the memory coming from the antibody side versus the memory coming from the T cell side would be a further complicating factor.
Yeah.
And if I'm not mistaken, natural immunity to papillomaviruses is not necessarily antibody driven. I think of it as more cellular. I think that's right.
I believe that's correct.
So this is, I think we've mentioned this before. This is vaccinologists fooling Mother Nature.
Mm-hmm.
They're inducing a mechanism of immunity that's not the normal mechanism of immunity, but is every bit as robust, if not more so.
So this was published last year, and so now we know that HPV vaccines prevent infection. They're probably the only one. It's the unique biology of the virus combined with some interesting immunological features.
Yeah.
You may have mentioned this, Alan, in your introduction, but this all comes from that evil institution, the National Institutes of Health.
Yes. Yes.
Okay. With help from elsewhere, like Fred Hutch.
Yeah.
And since I already did the work for measles, we will do that another time because there's
that'd be good.
Yes.
It's actually not as involved as this. It's simply an observational study where kids with antibodies get subclinical measles, right? They don't have a rash. They're fine. But if you look at, and this is interesting because it was done a while ago, they look at seroconversion, right? Because they don't have PCR. They don't do infectivity, but those kids seroconvert. But we'll do that.
Yeah, no,
Right.
I definitely want to read that paper.
So now we're moving to cancer.
Another cancer.
Another cancer and another vaccine that prevents cancer, actually.
Maybe. This is a preprint. This is a bioarchive preprint.
No, it's
in mice.
It's published in Cell.
Good. Okay, so it is out.
So yeah, the link
Cell.
in the show notes was to the preprint, but it came out in Cell very recently. And it's actually, technically it's in the May 14th issue of Cell, though it is online on Cell's website right now.
Right.
So we went to the future to read
Published
this paper.
online ahead of print. That's
Yes.
become the thing, yes.
Cell paper. It's called a short article, but gosh, it has a lot of figures.
There's so much data.
This is what it
Give me a
takes to get a
break.
short article in Cell now, yeah.
It is just so dense.
Yeah.
Respiratory viral infections prime accelerated lung cancer growth.
And the first co-first authors are Wei Qian and Xiaoqun Wei, and the senior author is Ji Sun. The team is from University of Virginia, Charlottesville, University of Pittsburgh, the Mayo Clinic in Jacksonville, Florida, and Cedars-Sinai Medical Center in Los Angeles.
So there are two viruses we're going to talk about in this paper, SARS-CoV-2 and influenza virus. And as you know, when some people who get COVID have lingering symptoms, which we call long COVID, and this happens with other respiratory infections, including influenza, that you have prolonged health issues, right? There have been some suggestions that SARS-CoV-2 infection is linked in some way to lung cancers, but it's the results are not consistent. Some studies report that if you were previously infected with SARS-CoV-2, you have an elevated risk of a variety of respiratory diseases, including lung cancer. And so why might that be? Well, when you get infection, especially long COVID, you have sustained inflammatory activities. This can lead to cancers, right? You get oxidative damage, oxidative stress, DNA damage, genomic instability. That can lead to cancers, can compromise immune surveillance. And so, but they say, we don't have a link between prior viral, say, pneumonia and cancer development.
Yeah. So just to point out, on episode 1243, we talked about another paper that sort of talked about respiratory infection and cancers. In that paper, they were looking at people who previously had breast cancer, and they showed data that respiratory infections, SARS-CoV-2 and flu, were associated with a recurrence of that breast cancer, but actually also a migration of it to the lung. And so I went back and looked at that paper in some detail in prep for this. So I will just note that in that paper, they were looking at people who had previously been diagnosed with cancer and their recurrence. And in this paper, it's they're looking specifically at new cancer diagnoses.
Right. And like that paper we have here, we're going to have some human data and then a lot of experimental data in mouse models. Now, they tell us that tumor-associated neutrophils, and that you have to remember TANs, I might use that, can be tumor-promoting or tumor-restraining, depending on a lot of different things. In particular, in lung adenocarcinoma, neutrophil-rich microenvironments correlate with poor prognosis and resistance to immune checkpoint blockade. And so, but how this works, we really don't understand. So what they're going to do in this paper is look at whether and how prior respiratory infections, viral infections, can reshape the lung environment to promote tumorigenesis. In fact, if I remember in that breast cancer paper, they also said that it was that paper. It was another paper where they said vaccination makes higher PD-1 levels on tumors, so that you might be a better candidate for immunotherapy.
Yes, so that was episode 1265, where they indicated that receiving an mRNA vaccine changed the immune microenvironment and thus perhaps made you a better candidate for cancer therapies. And so
what did we call that one?
That one was something like mRNA vaccines make cancer therapy great again.
Yes.
Something like that. So I linked both of these in the show notes because this seems to be sort of a general theme. I didn't spend as much time looking at the mRNA vaccine paper as the breast cancer paper when I was prepping.
This is a definite
we have an arc here.
Yes.
We now have an arc of
as I recall, we got a very intelligent email after the breast cancer paper that at least explored some nuances, shall we say, of that study.
Yeah,
but I
she
forget
said
the details.
the human data were not so great, but the mouse data, she said you would be better at the mouse data were very clear. Right?
Yes.
Okay.
But again, mice are not humans, right?
Right. Mice lie.
Yeah, but it does sort of seem like now that there are multiple studies that are sort of going on to this arc that there's perhaps something there.
Well, as you will see, we have some very clear mechanistic basis for this now.
I don't know if this is the right time, but I would like at some point, maybe now, for Brienne to give us the 1-minute introduction to neutrophils. I had to, there's a lot of cells running around, and I had to look up neutrophils to sort of refresh or prime my memory.
Yes. So neutrophils are innate immune cells that basically largely work by phagocytosis. So they're sort of the primary phagocytic cell. They are the most common white blood cell circulating in the blood in most people. So if you get a complete blood count, you'll see your neutrophil count, and that's usually the highest percentage. The cells are pretty short-lived, so they are present in the bone marrow, can be released from the bone marrow upon a variety of stimuli with infection. And then I often think of them as, the SWAT team that just comes in and phagocytoses out a bunch of whatever needs to be phagocytosed. There's some other cool mechanisms they can do, one called neutrophil nets. That's a bit newer.
I remember that. Yeah.
And so I've realized I shouldn't always disparage my neutrophils in not giving them as many functions as other cell types. But we usually think of them as sort of the key innate immune short-lived phagocyte.
I really like the SWAT team analogy.
Yeah.
They run to trouble and eat everything in sight.
Yeah.
Oh yeah. Part of what I read was that a wound or an inflammation where there's pus, the pus is mostly neutrophils.
Yes.
That's the white stuff, right?
Yes. The white stuff in your pus is neutrophils. It's really the dead neutrophils. So part of the idea is that they're short-lived. They're, eating something and then they're dying, which is kind of nice in constraining it. And as I tell my students on a slide where I talk about this in class, I once, when I made this slide, tried to have a picture of pus. And I recommend not doing a Google image search for pus.
Do not do a Google image search.
That's what I learned. And so my slide just says pus and has no image.
So the first part of this paper is a human study where they look at the relationship.
44,229,908 medical records.
Well, they start with 75.9 million adults,
yes.
Right? You were either uninfected, had mild to moderate COVID, or it's severe, requiring hospitalization. This is 2020, 2021. And then they looked at new cancer diagnosis from 2022 onward.
So the size of this database that they've got access to is immense.
Epic Cosmos.
Yeah. And I assume that's from Epic, which is one of the big, if not the biggest, EMR medical record systems.
Is this U.S. based?
Yeah. US based.
Wow. I'm surprised we have that big a database.
Oh, we have by law. Yeah. Everybody has to use electronic medical records now.
Okay.
And most places use one of a couple of vendors. And so that's
but there's not one universal thing. They don't talk to each other, right? Yeah.
They kind of.
But it's not like in the UK where you could get everybody.
It's not a nationalized registry, right?
Yeah. Right.
Oh, we wouldn't want to do that.
No.
But there's the Kaiser Permanente one, which has a lot of many millions of people too, and so you can get statistically powerful.
There's got to be a business where these are all amalgamated.
Oh yeah.
All right. So patients with severe COVID had a modest increase in overall cancer incidence, but prostate and female breast cancer was slightly reduced. Prior non-severe infection was associated with a moderate decrease in lung cancer diagnosis, whereas prior severe COVID-19 was associated with a higher lung cancer risk, 1.24-fold increase in the hazard after adjustment for comorbidities and so forth. Patients hospitalized for COVID had the highest cumulative lung cancer hazard compared with ambulatory and non-COVID controls. And prior severe infection remained associated with increased lung cancer risk after removing other comorbidities. So just in this human database, the suggestion is prior severe COVID is associated with increased lung cancer risk.
So if there's a hazard ratio of 1.2, does that mean 20% more likely? Can I do that?
I think of it as 1.2 times. So, yeah.
Okay.
Yeah, 20%. Daniel makes percentages all the time. Yeah.
Okay.
So if there's a 0.8 from a vaccine, it was a 20% reduction in cases by the vaccine. Yeah. All right. So now they go into animal models and they make animal models to look at this. They have a mouse model where they inject tumor cells intratracheally into the mice, right? They have KRAS and P53, KRAS mutant and P53 deficient lung tumor cells. They inject those into mice, right? And they're going to either establish or not. And so they infect them with SARS-CoV-2. They wait 2 weeks after viral clearance, 21 days, and then they inject these tumor cells into the mice, and they look at survival or tumor burden. Okay. So infection with SARS-CoV-2 had no impact on initial tumor development, but from week 3 onward, they had enhanced tumor outgrowth. They have reduced survival as a consequence. Okay, so it's infecting with SARS-CoV-2 in mice makes them have more tumor growth. They do the same thing with influenza A virus. They infect mice with that, and they challenge them with these tumor cells, and they also have increased tumor burden and increased mortality. And this effects persists 4 months after infection. So they can give them tumor cells at 4 months, and you see the same effect. So that's an orthotopic model where you're sticking tumor cells into mice. Now they have genetically engineered models where they have knocked in this KRAS, this altered KRAS gene, and the P53. They've floxed out the P53 gene. They've knocked it out. So, again, in influenza A virus infected mice, they have greater tumor growth and reduced survival. They use 2 different SARS-CoV-2 strains, and they also find the same thing. And they also have increased tumorigenesis in a lung cancer model where you put a carcinogen, urethane ethyl carbamate, induced lung cancer. And again, the prior infection makes it worse. So multiple approaches in 2 mouse models to say, 3 mouse models, that, if you have prior viral respiratory disease, it increases your tumor risk. So how can you prevent this? Maybe vaccination would be, and they state vaccination is one of the most effective approaches for preventing acute and chronic viral associated diseases. And I'm trying to think, what's better than vaccination? Oh, not leaving your house, right?
Yeah.
So they immunize mice with 2 doses of mRNA vaccine, spike encoding mRNA. And then they try the tumor burden experiment again, and they have substantially reduced tumor burden compared with the mock immunized mice. They also do the same thing with influenza virus. They give them an influenza virus vaccine, inactivated, and it reduces the tumor burden.
That was where things for me got really cool, because I was like, okay, so this is maybe some weird thing that SARS-CoV-2 does, and it's a particular case. And then you look at flu, totally different virus. Yeah.
I'll bet you this is generalizable.
And at that point,
adenovirus.
Yeah, they mentioned that they looked at bacterial pneumonia as well, and they said it sort of seemed to have increased things, but wasn't statistically significant.
Right.
So they don't look more broadly than that.
But the immune response against bacteria versus viruses is going to be different.
It's going to be different,
and that seems to be, as we're going to get to, that's a really critical component of this.
But the fact that they actually were able to show that vaccination helped is critical. And, more evidence on the massive pile of evidence supporting the importance of vaccination. And so that's one part that wasn't in the breast cancer paper.
Yeah.
They actually sort of were specifically trying to sort of look in the human data pre-vaccine release. And so it'll hopefully be cool in the future to sort of see if you can see the same effect of vaccination in that situation.
So they think the vaccine works because it prevents severe disease, and they can infect mice with low-dose SARS-CoV-2 or influenza virus, and you get moderate disease, and that doesn't increase the tumor burden. So it looks like getting the severe disease is the key. And we know that vaccines prevent severe disease, unless you're talking about HPV vaccines, in which case not only do they prevent disease, but they prevent infection. Okay, so how does this work? Let's do some RNA sequencing. They take whole lung cells from the mice, and they have it at 6 and 12 weeks after tumor induction, and they RNA sequence, right? They take all the cells, single cell RNA sequence. So teasing out single cells from these lungs from the mice, and they can tell from the RNAs that they detect what kinds of cells are there and what else is going on. And then they see airway and alveolar epithelial cells, and they can look at signatures of, and they find that they have tumor signatures, right? In particular, these alveolar cells and alveolar intermediate cells, they have, they're making RNAs that they say occupy a differentiation trajectory leading toward more transformed state. Okay. And so this is evidence that the infection is promoting tumor progression. You're seeing it in the RNAs that are expressed. So even though this sequencing reveals this reprogramming towards, transformed cell, the cells that they isolate from infected mice, they're not better at making organoids in culture. And in fact, the tumor number, size, and burden is comparable between PBS and influenza virus treated mice at the 6 hour, or is it this T6 time point? How many, is that 6 hours or 6 weeks? I don't remember. Anyway, so they say that means epithelial imprinting alone doesn't accelerate macroscopic tumor outgrowth. So it's not just these changes in the epithelial cells that's going on. Okay, so what else might be happening here? Well, there's a lot of immune cells in the lungs after infection, right? A lot of lymphocytes. There's myeloid cells. Hey, we talked about that last time. Myeloid cells, remember? Were you here, Brianne? Yes, you were.
I was. And neutrophils are a major type of myeloid cells.
And neutrophils, you got them in here. And so these are lung origins. These are neutrophils in the lung, not from the blood, right? They're not coming in from the blood. These alveolar, so they can do RNA-seq on these cells, right? The macrophages and the et cetera. And they show pro-tumor signatures. We're going to have to take their word for it because I don't know what a pro-tumor signature looks like in terms of RNA-seq. And I'm not going to go look it up. Okay. It would take like 6 months for me to do it. So they say what's happening here is that the local lung environment is being transformed into a pro-tumor state by virus, in fact, by viral pneumonia. Okay, so then they put the neutrophils into clusters, and they found that 0, 2, 3, 4, and 7 are present in non-tumor lungs, but clusters 1, 5, and 6 were highly enriched in tumor-bearing lungs. Okay, and this cluster 1, okay, in particular, is enriched for a gene called Siglec-F, which is a marker associated with pro-tumor neutrophils. And in fact, they do flow cytometry, and they can see that neutrophils that express high levels of Siglec-F are residing in the lung parenchyma. And so the idea is that previously infected tumor-bearing mice previously infected tumor-bearing mice that had been vaccinated don't have these Siglec-F high neutrophils compared with non-vaccinated mice. So Siglec-F seems to be important. And they do some pathway analysis, which means that they look at all the genes that are expressed, and they try and see whether what kind of pathways are involved in this. And they see pathways associated with recruiting myeloid cells, tumor proliferation, glycolysis. Tumors like to do glycolysis rather than oxidative respiration, hypoxia, immunosuppression. Those are cells that have high Siglec-F, and the low Siglec-F are linked to neutrophil activation, T and NK cell activation, and interferon signaling. So that's a hint there, right?
Yeah. So it seems like the infection has made the neutrophils go bad.
They made them go bad. That's right. So they want to do an experiment to see if infection-primed Siglec-F high tumor-associated neutrophils have tumor-promoting capacity. So they implant the tumor cells subcutaneously with either Siglec-F high or Siglec-F low neutrophils. Okay. if you put them with high, they get a bigger tumor mass than if you put them in with Siglec F low neutrophils. And they also took macrophages from tumor-bearing lungs that had been infected with the influenza A virus, and they also make an enhanced tumor growth. So they think that Siglec F is a driver of enhanced tumor growth after viral pneumonia. And this has been described by other people as well, this tumor-promoting neutrophil signature. So they conclude that prior viral infections program a pro-tumor lung microenvironment characterized by these Siglec F high tumor-associated neutrophils. Okay, is that clear to everybody?
Yep. Mm-hmm.
Yep.
Okay, so as Brianne said, we can just say the infection made them go bad, the neutrophils, and now they're promoting tumor growth, which goes to show you that the immune response is really complicated because it can't even keep things straight itself. Right?
Yes.
Are they trying to do something good and just going down the wrong
That's a good
pathway,
question.
or is this just?
I mean, I think it's hard to tell, but probably they are trying to do something good and are going down the wrong pathway. My guess is that this is super guessing. Is that perhaps these neutrophils are really good in helping you repair damage and repair a wound, but that also is a very proliferative type of situation. And so we might also be getting sort of hyperproliferation with a tumor because these neutrophils are trying to repair a wound and sort of support the proliferation of cancer cells as well.
I really like that guess. That's good.
Yeah.
It's a good way to think about it anyway.
But this immune microenvironment, pro-tumor versus anti-tumor, or, is obviously a big topic in oncology these days.
Yeah.
And the way I think of it is exactly as Brianne just stated it, that these are mechanisms these cells have for good evolutionary reasons, and that somehow they end up they get the wrong memo. And now we're doing this program over here, but that's not the right time to do it. And so it makes it a pro-tumor environment.
Okay, so they want to know, so neutrophils can be reprogrammed in the bone marrow. So they want to know if these bone marrow-derived neutrophils have this same property.
Yeah.
So this
is the same thing as last week. Are they local or are they coming from the bone marrow? And while I know about local macrophages, I didn't know as much about local neutrophils, so I was surprised to see this show up again.
All right, so they take bone marrow neutrophil progenitors and they transfer them into mice. They don't enhance tumor growth in this model. And so these pro-tumor effects are lung-specific. So they do a parabiosis experiment, just like last week, right? They link two mice together. They link their circulatory systems together.
Close your eyes.
Yeah. They have an uninfected mouse and a previously infected mouse, and they leave them there for 3 weeks, and they both get tumor cells, and they both get increased tumors.
Yeah, but the one that has influenza, so its lungs had this microenvironment, gets a higher tumor burden.
That's right. Yeah, the influenza virus pairs had more total and Siglec F high neutrophils than their PBS parabionts.
Yeah, so these neutrophils don't seem to be in circulation; they seem to be staying locally to the lung.
Right. So that's right. You put the two mice together, and they stay in the one mouse that was infected originally. Right.
Yeah.
So.
Yeah, so
these are resident neutrophils?
I guess so. I didn't know there was such a thing until I read this.
Well, same as we've talked about macrophages. There are resident alveolar macrophages, right?
Right, and they have their own developmental pathway. And
so where do they come from? Because they're short-lived, right?
The macrophages are longer lived than neutrophils.
Neutrophils, but where would new ones come from?
Where would new neutrophils come from?
Yeah. Don't know.
I learned in this paper they exist.
Yeah, they say prior infection creates a lung niche that locally drives accumulation and reprogramming of pro-tumor neutrophils. Okay, so the next they profile the cytokines and bronchoalveolar lavage fluid with these 2 mouse models. They have a genetic and orthotopic model, and they find that, upregulation of cytokines, GM-CSF, IL-6, and certain chemokines. On Immune last week, we had cytokines 101. That was a great episode. I learned all about that. These are factors that promote neutrophil development, recruitment, and survival. So it's a correlation there, right? So then they want to ask, does this increased cytokine response reflect epigenetic priming, right? This is the same thing did we talk about it last week? I think so.
Yep.
Yeah.
We talked about the method that is a sort of a high throughput method for looking at chromatin availability.
Right. We talked about trained immunity, I think.
Yes, it was last week as well. So the idea being that when chromatin is open, it's more readily transcribed. So if you leave it open, you can turn on genes right away. Whereas if it's closed, it takes longer for them. And here they're getting these cytokines turned on right away. So they're wondering if there's been some epigenetic modification. So they have a single cell assay for transposase accessible chromatin using sequencing. Oh my gosh. So if open chromatin can more readily receive transposons, and they can detect that by sequencing. So they do that on total lung cells from either SARS-CoV-2 infected or control mice. And they look at macrophages, epithelial cells, fibroblasts, and endothelial cells. They see mild to moderate increases in chromatin accessibility at the CSF3 promoter and enhancer regions. And they also see some other regions as well. What is CSF3? Are we going to learn that?
CSF is colony stimulating factor.
Yeah.
But I don't know CSF3 specifically.
So they don't actually talk about what they think it does. But their bottom line is that if you get infected, you get epigenetic remodeling of cytokine and cytokine loci in immune and structural cells as well in the lung. And they say total lung cells from virus-infected mice make more GCSF when you stimulate them in vitro, and purified macrophages, epithelial cells, and fibroblasts also produce more GCSF. So it suggests that the chromatin is opened up already, right? Some kind of trained immunity. All right, so a couple of more experiments here, actually quite interesting. So SIGLEC-F can be induced by GM-CSF. How something's coming around here, and TGF-beta. This is GM. I'm confusing GCSF with GM. So GCSF is different.
And there's also just CSF.
Yeah, right. Anyway, so GCSF we see is already up, but it's been previously shown that SIGLEC-F can be induced by GM-CSF, but neither was upregulated in their prior infection. So they took GCSF and they find in vitro that can induce SIGLEC-F expression, and together with TGF-beta, it makes it even higher. So what they did is they gave mice intranasally GCSF plus TGF-beta. This enhances neutrophil recruitment and SIGLEC-F expression. And when they treat the mice in this way, it augments tumor growth. So these cytokines, GCSF and TGF, when given to mice, augment tumor growth. If you block GCSF signaling, which happens after GCSF binds its receptor, it reduces these SIGLEC-F high neutrophils and it decreases tumor burden. So what does this all mean? Prior respiratory virus infection, it imprints changes in the lung microenvironment that promote neutrophil recruitment and reprogramming, and that creates a niche for tumor growth. And you can prove that with just GCSF and TGF-beta by giving that to the mice. So you don't have to rely on infection-driven epigenetic reprogramming. So doesn't that suggest strategies for therapeutic intervention?
Yeah.
It does.
So now they're going to target neutrophils.
This is the point in the paper where I said, okay, now y'all are just showing off. You had enough data for the paper already, but now you're just going to...
So, Brianne, they're using antibody to LY6G. Is that going to target neutrophils?
That is going to target neutrophils.
Okay. And then they have, they can blockade chemokine receptor 2 signaling with a drug called riparixin before tumor inoculation. So they infect their mice and then they put the tumor cells in. But before they do that, they do these anti-neutrophil activities. And these partially reduce neutrophils in the lung and they decrease tumor burden. They get improved survival. It's not 100%, but it goes down. So it's good. So now PD-L1, right, which is immunosuppressive, right, and it is upregulated in virus-infected lungs, which suggests that this tumor microenvironment after virus infection is increasing PD-L1, which is mediating inhibition of T cell responses, right, because it's binding to the receptor on T cells, and then the T cells won't kill the tumor cell. So they tested whether their neutrophil inhibition plus PD-L1 blockade could be useful. So they take SARS-CoV-2 infected tumor-bearing mice. They treat them with the anti-neutrophil drug. They treat them with anti-PD-L1 or a combination of both. And the combination significantly suppresses tumor burden. So you blockade neutrophil driven immunosuppression, T cell checkpoint inhibition, which is what the PD-L1 is, and then you can start to counter, at least in mice.
Yes.
But I like the way you go from the observation, the clinical observation. You go in a mouse model, you make the same observation, you find that it's due to a certain population of neutrophils, which have gone wrong, and then you can design some therapeutics. It's pretty nice.
So my understanding could be, not totally right, but my understanding is that viral infection influences the cells of the lung and changes the cytokines being made in the cells of the lung, sort of changing chromatin accessibility in those lung cells so they're able to make cytokines well. This cytokine change either makes it so that neutrophils that are already in the lung go bad, or that any neutrophil that happens to come to the lung goes bad.
Mm-hmm.
And this neutrophils going bad thing helps make this environment a good place for a tumor.
Yeah.
And somehow also in this, we are turning off T cells, but exactly how the T cells are getting turned off is not directly clear.
I wonder if you could use material from surgeries on humans or autopsy material to look at the microenvironment. I mean, I would think that the bad newts would be around in lung cancer patients.
Probably, yes.
Mm-hmm. Yeah, they said that in the beginning.
Yeah.
That, adenocarcinomas have these newts, these bad newts. Yeah.
Okay.
But the thing is, you have to have a certain percentage of PD-L1 on your tumor. Otherwise, they're not going to give you this anti-PD-L1, right? Because it's not going to make any difference. And that's why the COVID vaccine was cool, because the idea was that bumped up PD-L1 on your tumors.
All the more reason to get vaccinated.
Yeah.
So that's the thing here that, well, yes, you should be vaccinated, then this won't happen. But if it happens, this combination is nice, but they're not going to do it unless your PD-L1 is, I would have to do clinical trials to see the response. Right?
Yeah.
That's the bottom line here. I mean, that's it. So they say that, this is probably a general thing with viral infections. Viral infections are the most prevalent lung insult. And so they think it's probably even wider spread than we know. And given all the people infected during COVID, in the next years, it could be reflected in a big increase in these cancers. So they say we can target these Siglec-F high neutrophils is challenging because Siglec-F is shared with other lung populations as well. So that's not great. So we need to do more work. Requires further study.
Yes.
But I think it's pretty interesting, right?
Yeah, and we don't know how long this pro-tumor environment, the neutrophils holding out the sign saying tumors settle here. We don't know how long that persists after the severe infection, but clearly it seems to be long enough that if you're going to start developing a tumor during that window, this increases your risk. And to remind people, we're all generating cancer all the time. This is just something that, as a part of being multicellular, some cells occasionally decide to start multiplying out of control, and then the immune system comes by and blows the whistle and takes them out. And that's the normal process, and it's when that process breaks down, like these neutrophils going bad, that you then have problems. So you need both of those things to happen. You need the bad neutrophils, and then you need some sort of event, but it's fairly common, that starts the cancer going.
So is it a 20% increase in cancer risk in severe COVID patients? It's a lot.
It is.
Yeah.
I don't know what the timeline is either.
But people who developed severe COVID, certainly early in the pandemic, they were sick for a long time. They were spending a long time in the ICU and then very slowly recovering afterward if they survived.
Well, look, folks, if you don't want to get lung cancer after respiratory virus infection, just get a vaccine.
Yeah.
And don't listen to the anti-vaxxers who are spewing nonsense. None of what they say is correct.
And your flu shot counts too.
So, there's this one trope, anti-vaxxer trope that's going around, which says, the COVID vaccine killed thousands of people, because there were excess deaths during COVID that we don't quite know where they came from. So Daniel did a study, reviewed a study on Clinical Update yesterday. And it turns out that if you were vaccinated, your excess death risk went down, not up. So it protected you against excess mortality. These people are just, the more I think about it, the crazier I get, because these are proven things. For years, vaccines have been proven to work and prevent people from remaining non-adults.
Yeah.
Unfortunately,
I don't get it.
Unfortunately, we're living in an environment where truth, it's such a lying environment, okay, that it gets very confusing for people who are not.
It's worse than
thinkers.
It's worse than a Siglec-F Heim environment.
It is. Part of me feels like we need, some influencer on Instagram to be like, let me tell you about this new wellness hack. It's called a vaccine, where you just give. What if I actually just took a small dose of virus that was mutated so it couldn't make me sick, and I trained my immune system? That's my new wellness hack.
I use a homeopathic remedy to prevent flu. A little bit of diluted flu before the season.
A little bit of inactivated.
A little bit of inactive flu, yeah, and it's just a tiny bit. That'll set you up.
Let's do a couple of emails. Brianne, can you take that first one?
Sure. John writes, Doctors TWiV, I'm sure you've heard that Bhattacharya has a lecture series at NIH that is to unimaginable astonishment titled Freedom from Science. Jesus, haploid Christ. How did we get to this point, and how will we ever get back? You've probably also at least heard that Angela Rasmussen's takedown of the first lecture on COVID origins, imagine that, but it's 4.5 hours long. I gather it didn't go smoothly for Matt Ridley, but I've just started watching it and we'll have to take it in chunks. And he gives the link. I also am taking that in chunks myself. Anyway, in haste, on the remote chance this is news to you, here in Greater Braddock, we had 84 degrees Fahrenheit Sunday afternoon and 22 Fahrenheit early this Tuesday morning. I guess that swings like that are common in deserts, but we had a thunderstorm in between.
Why is Matt Ridley now posing about this COVID origins crap? What does he know?
He wrote a book.
I didn't realize that the H was for haploid. That's pretty good.
I
I
like
was
that
glad
too.
to see somebody else was aware of
It's
what the
good.
middle initial was.
Alan?
Sure. John, again, writes, Doctors TWiV, especially Kathy. Continuing from my last one, re BO, it crossed my window by total coincidence that there's a lab at Karolinska that focuses on olfaction. I remember some years back that anosmia was frequently mentioned in connection with COVID. This lab may not have started on olfaction when COVID hit, but they were active in working on the connection between the two. But the thing that could be a pick, if I could find an underlying paper, is that they've apparently figured out how to transmit certain smells via internet. For that teaser, search this link for Rose, not Nose, provides a link. Otherwise, another thing to follow regarding SARS-CoV-2 origins, a lecture series at NIH that I imagine you're already on top of. Here in Greater Braddock, we had 84 Fahrenheit Sunday afternoon and 22. Oh, that's the same. It's the same day. So 22 after early as Tuesday morning. Yeah.
All right, let me take the next short one from Jim. He provides a link to a paper in Cell Host and Microbe. Commensal enteric virome regulates intestinal carbohydrate digestion and absorption. And he writes, studies suggest a beneficial role of the gut virome. I think I recall Vincent once saying viruses might play beneficial roles in humans, but none have been shown. This seems to make that closer. Yeah, so these are mainly phages that are doing this, right? Because they're influencing the bacteriome, the microbiome. And I would like to find eukaryotic viruses that are beneficial. But yes, this is good. I saw this paper and I don't do phage papers on TWiV because we have a resident phage person here. All right, Rich, can you take that last one? No, there's one.
There are two more.
One. Two more, yeah.
Right. Robert writes, hello, TWiV. I was listening to TWiV 1303, Overachieving Vaccines, and caught the discussion about the costs of using Google's language AI Gemini to answer questions. It turns out there's nearly as much misinformation and uncritical assumption about this topic as some others you discuss. Google has published a detailed technical paper and blog summary about the resource cost of their AI inference. Inference must be interface.
Yeah, it's inference.
Inference.
Okay, inference. Okay, which is what you're doing when you ask Gemini a question. They estimate it at about 0.24 watt-hours of electricity and 0.26 milliliters of water, or the equivalent of less than 9 seconds of watching TV. I have to send this stuff to my son. I think this is part of his job. What he's been doing for the past several years is to figure out the carbon footprint of various data centers and stuff, and for Google clients, try and help them understand the most carbon, minimize their carbon footprint in how they parse their data analysis. I need to send this to him. At any rate, sorry, little digression. Okay. Vincent's observation that he might release more carbon spending time to find an answer with conventional services versus asking Gemini is also probably correct. There's a decent amount of research on the topic, and for tasks AI systems perform well at, they are typically more efficient, often much more so than either humans or conventional computer algorithms performing the same task. In my own field of medical imaging, I have seen common tasks such as identifying brain structures in images go from laborious manual job taking hours or days per scan when I was a grad student, guess who got to do this, to a mostly automatic task taking a few minutes to a few hours using conventional algorithms to something that takes a fraction of a second using modern AI. That's all running locally on your laptop too, no cloud obfuscation, and where you would certainly notice megawatts of power and untold liters of water being dissipated into your thighs. The claims about exorbitant water use seem to trace a tortuous path back to original, quote, sources that are often vague or just wrong. Another common error is conflating the amount of water needed for a one-time fill of a closed-loop cooling system like your car's radiator with water that is continuously consumed. AI inference has also become much more efficient since the ancient days of the original ChatGPT through better hardware and especially better algorithms. None of these things are free, including at 9 seconds of TV, but we should all keep in mind, and we should all keep that in mind. But some of them might just cost a few orders of magnitude less than you might have heard on the internet. Rob from Toronto. Thank you, Rob.
Point taken, but I would point out, if these AI models were so darned efficient, where is all the enormous amount of electricity going that's requiring building entirely new substations and generating plants and jacking up everybody's electric bills in areas where data centers are being built? It's going somewhere.
No, the electricity use is substantial.
It is significant, and
especially in training the AI.
What occurred to me reading this email is two things. First of all, the training, which I don't know if Google is including that in their tally of 9 seconds of TV. That's maybe what the final model takes, but you need to internalize the cost of what it took you to get there. But the other thing is, it may very well be that text-based queries like you're doing, Vincent, would probably be much lower energy than the AI video porn that people are generating, which is probably using a lot more of the power.
Or programming, right? Writing a program.
Or writing a program, although that's also a text-based query.
So I've been reading lately a lot that it's not even clear that the data center approach is the right approach.
Right.
Right? You could do it locally. It's just that these companies don't want to share their libraries with you, right? Because they want to make money on it. But it's not really appropriate for them to push it on governments and to tell you that you're going to fall behind if you don't build a data center, because I don't think it's been proven that that's the way to go.
There's also a tremendous amount of conflation, convenient conflation of different types of things that are being called AI.
Yeah.
So the algorithm that's running on your laptop to do one of these radiology things is not what you would call AI if you're talking about ChatGPT or Stable Diffusion. It's doing a different thing, and it's
machine learning.
It's really more like a souped-up version of the previous algorithms you used to use, because otherwise it couldn't run on your laptop. But so there's some sleight of hand going on with that. But point taken, I do not have the full data set to impugn your use of AI.
These data centers are mainly running on GPUs, right?
Yes.
Because they're really good at doing things repetitively, right? They were made for gaming, and it turns out they're perfect for this. But, Apple now has made this new M5 chip, which is apparently amazing and is going to be able to do all this locally because, the GPU and the CPU and the neural engine are all on one chip. Whereas in these data centers, right, we're running thousands and thousands of separate GPUs. So we'll see what happens. I don't think we're there yet. I think the end result is yet to be seen.
Yep.
Okay, let's do some picks. Brienne, what do you have for us?
So I have a pick that is in some ways repeats of previous picks, but I felt like it was important. So I think that both Alan and I have previously picked the book Project Hail Mary by Andy Weir, which is just a lovely, lovely book. One of my favorites. That book was made into a movie which came out last week. And so I have, of course, seen said movie, and I have picked a review of said movie, which does have a section with some spoilers, which that you may or may not want to avoid depending on your feelings about the movie and spoilers and things like that. But I did really like this review because it highlighted a lot of the things that I absolutely adored about this movie. And I did feel like this was a movie about science that had a lot of heart and a lot of humor. And to me, a big part of it involved thinking about, friendships, or as it talks about here, collaboration in your network. I found this just to be a delight of a movie. So I would strongly recommend everyone see it. I saw it on IMAX, and seeing it on IMAX was also quite nice. So highly, highly, highly recommend.
What's IMAX, a kind of theater?
IMAX is a kind of theater with particularly large screens. There's usually pretty good sound. And so some of the scenes in this look very nice on IMAX.
You know, when I was a kid, the movie companies came up with this gimmick to try and compete with TV. What was it called? Cinematic, or, you know, they had really big screens. What was it called? Let me look it up.
Yeah, so I don't know if anyone else has seen this movie, but I absolutely was delighted by it.
I have not seen it yet.
I have not. I don't go to enough movies, but this one, I got to see if I can scare up some interest in my family. Either that or I'll just go by myself. I read the book a long time ago, and I'd like to see this.
Cinerama. Remember Cinerama?
Cinerama.
Introduced in the 50s, a deeply curved, massive screen.
Yes, I remember that.
To try and combat TV.
Yeah.
Everybody's got to combat each other. Rich, what do you have for us?
Okay, this is another strictly feel-good pick. It was an article in the Washington Post, I think, today. The title tells the whole tale. After a decade as a Yale hospital janitor, she is now a doctor there. And this is the story of a woman, comes from, single mother family of 3, with no advantages whatsoever. Nobody in her family had ever been to college. She had no idea, kind of what college was or how to apply. When she was 18, she needed a job, not only just for her own income, but to help support the family, because there were issues there. The classic story. And so she went to Yale New Haven Hospital and became a janitor. And she was a janitor there for 10 years, and in the process became interested in medicine. And so sort of bootstrapped her way through a college degree and some extra stuff to pump up her application, her CV. She ran across a teacher who served as a mentor to help her get through the whole process. Went to medical school at Howard and applied to Yale for a residency in anesthesiology.
And got in.
And just got in.
That's great.
Just matched for the residency.
That's awesome.
Great story.
Love it.
That's great.
Alan, what do you have?
So I have an article, news article from Science about a research study. You can follow links to the study, but there's also an interview in this article of the researchers who both became interested in the topic of jokes at scientific conferences. And I think we've all had the experience. You're sitting through a talk that's kind of dull, and the speaker cracks a joke, and maybe it's not all that funny, but it kind of brings you back to the subject, and gee, is that helpful? And how often does that happen? And are the jokes any good? And so they did a study and went through, categorizing jokes and figuring out who's telling them and when, and found that more senior researchers are more likely to joke. Male presenters are more likely to joke than female. But when people do fit a joke into the talk, it does seem to help in communicating the topic. And I really liked the idea of some serious research on this topic.
That's great. I try and tell a joke every class.
Good.
They kind of giggle, but I don't know.
I always found that, prepared jokes never worked. The only humor that worked for me was spontaneous. Yeah.
My favorite part of this, the interview was the phrase or the sentence, "We also assessed joke success," which was not easy to standardize.
Yes.
So, and you guys got a couple of jokes in here. They're pretty good. Where'd you come up with those?
Oh, I just came up with that off the top of my head after I saw Vincent's.
I just found it on the internet.
Okay.
My pick is a video by...
Are we going to tell our jokes?
Yeah, you should.
Oh, you got to tell your jokes.
Yeah. Okay.
I'd tell you a coronavirus joke, but it would take 2 weeks to get it.
I heard a herpes virus joke years ago, and it was so good I still sometimes erupt in laughter about it.
You have to be a TWiV listener to get that.
Yes.
I like those.
Yeah, they're good.
My pick is a video by Yuval Noah Harari. Why advanced societies fall for mass delusion. So he's a history professor in at the University of Hebrew. No, where is he? In Israel. Where the heck? Which university? Weitzman, Tel Aviv? I don't remember. I'm sorry. But you are a professor in Israel, and he's written a couple of interesting books. One of them, Sapiens. Now he has one called Nexus. And he's talking about the rise of information in our societies. And now we're in a phase where all the information is inorganic, right, by computers. And he spends a little bit of time talking about science, which is really good because he really likes science. And I find it really interesting. You should listen to it. And there's a comment in the comment section, which I read while I was watching it. It says, science works because it's suspicious of itself. Every claim is temporary. Every conclusion is on probation. In a world flooded with confident nonsense, that self-doubt might be our most reliable form of wisdom. That's a really good comment. I like that.
Yeah. And I just looked him up. He's a professor of history at Hebrew University of Jerusalem.
Hebrew University. Thank you very much. Anyway, I really like it. It's a long video. It's an hour. So you need to like long form stuff, and most people don't these days. But I really like his logic. It's really interesting to listen to. We have a listener pick from Greg. My suggested pick of the week is a recent paper in the new journal Oceans, Land, and Atmosphere Research, titled Changes in Terrestrial N Budgets and Riverine Nitrate N Yields from Mississippi River Water Basin Watersheds 2000 to 2020. Nitrate loads in the Mississippi River increased after 1960 with the expanded use of industrially produced ammonia fertilizer produced using the Haber-Bosch process. This contributed to increased algal production, eutrophication in the northern Gulf, and an extensive area of hypoxia in the bottom waters, popularly known as a dead zone, similar to other coastal waters around the world that have become similarly enriched. The problem was extensively studied in the late 1990s and early 2000s. NNP reduction goals were set, and monitoring continued. After nearly 2 decades with little sign of progress, nitrogen loads appear to have decreased and achieved an interim goal of 20% reduction by 2025. The recent article in OLAR uses a large-scale input-output analysis of biologically reactive N developed at Cornell, referred to as net anthropogenic N inputs, or NANI for short. The correlations between values of NANI and riverine nitrate water loads at 217 sites suggest that the decline in nitrate loads in the river is partly due to more efficient use of N fertilizer for crop production, mainly corn, and reduced deposition of oxidized N from the atmosphere, the latter the result of Clean Air Act mandated reductions in NOx emissions from smokestacks and vehicular tailpipes. I think it's nice to see progress occurring on this scale issue, even though it took many years of effort and consistent monitoring to see the results. The research was made possible by monitoring efforts of the U.S. Geological Survey, the U.S. Department of Agriculture, NOAA, and various state agencies that routinely monitor water quality. Additional funding was provided by the Walton Family Foundation to the Great Lakes to Gulf Virtual Observatory. Achieving the reduced nitrate loads in the Gulf was likely due to the efforts of farmers, crop advisors, and crop scientists in improving the management of N fertilizer for crop production. The article is somewhat jargon-laden, but the University of Illinois prepared a press release, which he links to, that provides some explanation for a general audience. Best regards, Greg in Champaign, Illinois, currently 79F and expecting thunderstorms with possibly damaging wind, hail, and tornadoes. Thank you for that. I appreciate it.
Yeah.
Except, all of this climate change stuff is being trashed, right? Clean Air Act, hasn't that been trashed?
Yep.
Yes. One of the nice things about this, I haven't read this paper yet, but it sounds like from this description, a lot of the progress is actually being driven by the farmers themselves. And this is not so much government coming in heavy-handed and saying, thou shalt not. It's things like extension service agents coming out and saying, hey, half the fertilizer you're paying for and putting on your field is washing down the river and not doing you a damn bit of good. Here are some ways to prevent that. So it's good for the farmers to waste less fertilizer. And that has the effect of also shrinking the dead zone, which is putting the shrimpers out of business down in the Gulf.
Right.
I just wondered if the monitoring is really going to go down now because
monitoring will go down, yeah.
Yeah. That's TWiV 1309. You can find the show notes at microbe.tv/TWiV. You can send us your questions and comments and picks of the week to TWiV@microbe.tv. And if you enjoy these programs, we'd love your support. Microbe.tv/contribute. Brianne Barker is at Drew University, BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
Rich Conda is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
Thanks. It's always a pleasure.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.